Okay, so can you hear me? Okay, sorry. Thank you, everyone. Thank you. I want to say thank you to the organizers for such a wonderful conference. It's been really great to meet many of my references, to know that they are not robots that write all day, but they are real human beings. So as Gerardo said, I'm going to present part of the results of my ongoing master's degree. And as you can see, we are many people who are working on this and other projects. So Barkeria is a genus consisting of 17 species, mainly found along the Pacific coast from northern Mexico to eastern Panama. And something interesting is that it grows especially in tropical dry forests. Many species are found in a habitat we may call as twig epiphytes. Some others may grow in rocks abundantly. Some others may grow even on cacti. Barkeria is one of the few orchid genera that can grow like this, while a few others grow in more humid environments like Barkeria skinneriae in tropical oak forests. And it's interesting that usually these changes in life forms and habits are related to contrasting life cycles. Also, in the genus, we find a couple different floral motifs, and the plants go from small, thick cespitous pseudobulbs to cespitous canes to scandent canes in which new stems originate a couple nodes above the origin of the previous stem, thus giving a climbing habit. And thanks to the floral morphology, in particular of the column, the adnation of the column to the lip, and the overall shape of the plant, Barkeria species were thought to be related to Epidendrum since 100 years ago. And this was confirmed by the very early ITS analysis performed by Vandermeer and colleagues. And this showed that Barkeria actually belongs to a clade within Laeliinae subtribe that is called the Epidendrum alliance, together with other genera like Colarthron and Orchidaceae. Similar results were retrieved with the analysis of both ITS and a few plastic markers, and this included Myrmecophila as part of the Epidendrum alliance. Using a very similar set of markers and a wider sampling of Barkeria species, Angulo and colleagues recovered Barkeria as a monophyletic group, with Microepidendrum subvolutifolium as its sister lineage. However, a couple nodes here within Barkeria were not highly resolved, and this analysis only includes one species of Epidendrum. This is important because the analysis of nuclear of plastidial markers alone recovered very similar results, but the analysis of the nuclear ITS only recovered Barkeria into separate clades. It's polyphyletic, and within several other species of the Epidendrum alliance. As we can see, as we have seen in many conferences now, the use of phylogenomic data, especially from chloroplasts, have helped to resolve the phylogenetic relationships within orchids from the family level to specific subtribes to particular genera. Thus, in this project, we aimed to assemble and describe the plastic genomes of Barkeria species from new generation sequencing data, then to infer the phylogenetic relationships within Barkeria and among closely related genera, and to explore the variation of selected traits under a robust phylogenetic frame. In particular, I'm going to talk a little about the evolution in the variation in genome size and chromosome numbers. So for this, we sampled for high throughput sequencing 15 species of Barkeria, given the initial unavailability of aperitif species, as well as Epidendrum species from different clades of the giant Epidendrum, as well as other species in the Epidendrum alliance of Microepidendrum, Colarthron, Orchidaceae, and Myrmecophila, as well as a couple other species from the Laeliinae subtribe. We complemented our matrix with a few plastic markers for Barkeria lindleyana and Europani, that were the plants that we were missing initially. And for sequencing, we used a hyb-seq strategy that comprises both hybrid enrichment with genome skimming. We use this strategy to gather hundreds of nuclear genes, as well as to increase the probability of getting whole plastoms and mitochondrial genomes, ideally. The pipeline to analyze only the chloroplast genome was to check the quality control in FastQC. In Trigonomatic, we filtered low-quality data and Illumina adapters. We used the de novo strategy to assemble plastoms using get organelle. And in Genus, we first transferred automatically the annotation of Masdevallia coccinea, which is from Peralta, that is the sister clade to Laeliinae. And then we manually expected all the annotations to verify stop codons and correct reading frames. We aligned everything with mapped, both coding and non-coding sequences. And we performed a phylogenetic analysis under maximum likelihood. Also, for estimating nuclear genome sizes, we used flow cytometry from ovary and pollinia tissues. And this was to avoid pseudo-replication, something that some people have talked before. And for further details, please attend the talk by my colleague, Rubí Meza, who is going to talk extensively on flow cytometry for orchids. And to make chromosome preparations, we used very classic and standard methods using root tips. So as a result, we could assemble 14 whole plastoms of Barkeria species. Then the only one we couldn't assemble was Barkeria melanocolum because the sequences were from a previous sequencing event, which only involved the hybrid strategy. And the whole plastoms that were assembled contained 73 protein-coding genes, 30 tRNAs, and 4 ribosomal RNA genes. In general, the plastoms did not vary too much in length, from 144,000 base pairs in Barkeria nevosa to 149,000 base pairs in Barkeria niflora, and the percentage of GC was actually very constant. In terms of the structure, actually, the plastoms were kind of boring because they are quite collinear, and we didn't find any particular gene inversions or corrections in the order. As well, the limits among the different regions of the plastom were very similar. But we found interesting variation in the presence of NDH genes. So as you can see in Arpophyllum giganteum, the NDHB gene is actually functional. You have an intron and 2 CDS, right? But what happens in species like Barkeria barkeriola is that we have a stop codon just a few hundred bases inside the gene. And in other species, as you can see here, we have large deletions. And in general, in many species of Laeliinae, these 5 genes of the NDH family are absent. And in Barkeria, we found only these 6. Thus, we didn't use NDH to make our phylogeny. And the alignment of whole plastoms, as well as the Sanger sequences, retrieved Barkeria as monophyletic with high support. And according to this result, Myrmecophila is a sister genus to Barkeria, and Micropedendrum is sister to this clade. On the other hand, Colarthron and Orleanitia are sister to Epidendrum. Within Barkeria, we recognized 3 main clades. We are missing a color here. Well, anyway, the first one is the Uniflora clade that is found mainly in the Pacific coast of northwestern Mexico. And the species in this clade are recognized by these whitish lips with highly contrasting blotches, similar pattern in the column. And the plants are this cane-like structure. All of them are epiphytes. And in the next clade, we find clades. The first one is the Barkeria ovovata clade, consisting of species with very small flowers in dense inflorescences and very small cespitous plants. Most of the species are found only in western Mexico. Out of only one species, Barkeria ovovata, reaches Costa Rica and Panama. And in the third clade, we find the scandent species of the genus. As you can see, species like Barkeria vaneriana are really climbers. And interestingly, we find in this clade species, species that are found only in the western side of the isthmus of the Tehuantepec, and other species that are only found in the other side. This is important because we didn't retrieve these species as monophyletic, and more important, one of them is actually sister to Barkeria whartoniana, which is a micro-endemic that even has preference for cacti, showing that probably the biographical and ecological history of the genus is more complex than what we expected. So in this phylogeny, using only whole plastoms, we coded the presence and absence of NDH genes. And it's very interesting to see that, in particular in these clades, we find a lot of large deletions as well as complete deletions of particular NDH genes. This is relevant because it's been hypothesized that the absence or loss of these genes may be related to the capacity of growing in highly illuminated, even perturbed areas. And as you can see, one example of that, this is Barkeria ovovata. This plant grows in very altered environments. This one is even a road. And if you want to see this plant, you can go like 400 meters away from this conference center, and you will find a very interesting population of Barkeria ovovata. So if you want to see it, please tell me after this talk. And for the genome size, we traced the values that we found, and it was very interesting to see that in Barkeria and the Barkeria uniflora clade, we find these very large genomes, contrasting with the rather small genome sizes found in all the other species. The variation is up to 5-fold if you compare Barkeria palmeri with Barkeria barkeriola, and it's very interesting to see. No spoilers. It's very interesting to see that here in Barkeria vaneriana, which is the hungry climber, we found that this kind of duplication of genome size. So we thought, is it a polyploid or something like that? And the answer is yes, it is. All the species marked in red, this is a character optimization, and all the species in the red clade have an haploid number of 36 chromosomes. For example, we have a nucleus of Barkeria ovovata, but suddenly in Barkeria vaneriana, we have 72 chromosomes, meaning that it's actually a polyploid species. On the other hand, the uniflora clade the species in the uniflora clade have 40 chromosomes, so we found a disploidy, and it's very interesting because these chromosomes are not only more numerous, but are also big. So mainly that's where all this increasing genome size is found. And yeah, so our conclusions are that Plastom of Barkeria are very consistent and vary mostly in the presence of NDH genes, and this may be related to the different ecological strategies inside the genus. And the alignment of whole plastoms retrieved Myrmecophila as sister to Barkeria and resolved the relationships inside the genus with high support. Changes in chromosome numbers and genome size support clades inside the genus, although increases in genome size are not proportionally correlated with increases in chromosome numbers, and Barkeria vaneriana is likely a tetraploid species. So I want to thank all the institutions that have supported this work. In particular, I want to thank the MONOCOTS committee for giving me an assistance scholarship to come here. And most of all, I want to thank my colleagues at Salazar and Granados lab. All these guys are really talented and amazing people that have trained me in so many ways. And if you ever want to come to Mexico and have subtitles and teach techniques to us, we will be very grateful. So that's it. Thank you.
Thank you. We have time for questions.
Thank you, Cekouat, for the wonderful talk. I'm very curious about this sister relationship between Myrmecophylla and Barkeria, because at least superficially, the vegetative and floral characters appear very different. But so I wonder if you have identified morphological synapomorphisms, possible morphological synapomorphisms between for that clade.
Yeah. The short answer is no, I haven't. And actually, this position is very interesting because, both Myrmecophylla and Colarthron have these shallow self-bulbs that are the entrance for ants. But other than that, it's been really hard to me to find other, floral synapomorphisms. But, and this is a spoiler, according to the astrophylogeny of all the 351 nuclear loci, the position of Myrmecophylla changes. So I think that we may be witnessing complex phenomena as incomplete linear sorting. Out of Gibnish already told us to not say this, but to address it. So that's what I plan to do. So yeah, it's an open question still.
Another.
Thank you for the great presentation.
Thank you.
You said, I think, that it's rare that the orchids are epiphytes on cacti. Can you explain why? I mean,
well, we have. Well, first of all, it's very rare that orchids grow in environments where cacti are abundant, right? Most orchids depend on high humidity levels, and that's why we find a high diversity in orchids in in cloud and humid environments, right? So, in particular, in Mexico, we have very large extensions of tropical dry forests, and some other genera like Encyclia or Catastines subtribe are very present in these environments. In particular, cacti are very interesting because they have these very smooth surfaces. So if one thinks about the small seeds of orchids, they usually tend to be or to fall on the surface of very rugose bark trees, tree bark. Right? So it's very rare to find orchids on these substrates, mostly because of that reason. But we have found that this species, Barkeria whartoniana, actually has a keen preference for these kind of phorophytes. We have quantified this, and this is one of two most favorite phorophytes. And the other one is Plumeria rubra, which is found outside this conference center, which also has this very smooth bark. So we don't really understand what this species is looking for, because it also lives in an environment that is very, very windy. You have this high-speed air currents happening all the time. So actually, I don't know. It just likes being. It just likes instability, I think, for reasons unknown to me.
Thank you. Cool.
Another question.
Hey, amazing, love it.
Thank you.
And what about the genome size distribution? And there are some correlation with latitudes. You know that higher genome size are correlated or not, but there are some papers that are talking that they are correlated to higher latitude.
Yeah.
Do you see something like this in your data?
To be honest, not really, because the species in these two clades, usually grow in very similar environments, even very close to the sea. On the other hand, these lithophytic species that grow in higher elevation sites have smaller genome sizes. So that's actually we are working on, trying to find other latitude. Oh, sorry. Oh, well, it may be. Interesting questions. I think I'm gonna look deeper into that. A first impression would be that maybe because the species in this clade are also found here in Central America, but the species in this clade are only found in northwestern Mexico. So it actually maybe. Thank you.
We have time for another.
Hi. Why did you choose the angiosperms 393?
353.
So sorry. I think it was not 353, right? So I was wondering why specifically did you pick that one? You know, Oscar also other people were all using angiosperms 353. So I was wondering what was the.
Well, basically because of the collaboration history of my life. They started this collaboration many years ago with the lemon, but probably my advisor, Gerardo Salazar, can go deeper into that.
Okay.
Yes, thank you. When we received the funding for this project, it was at the beginning of the pandemics, and the 353 kit was very new, and we had to make decisions. And upon checking the results of the several analyses published, the poorest recovery of the genes was for Asparagales, and orchids are Asparagales. No orchid was analyzed, but the Asparagales present had very poor recovery, so we had to make a decision, and we already had experience recovering 300 genes from the lemons kit, so that was a practical decision. But let me tell you that a member of Dr. Granados' lab, we work in collaboration with bromeliads and we with orchids, have found some important overlap between the two kits. I think they found about 80 genes that overlap between the two kits. So eventually we may be able to put all information together. Thank you for your question.
Okay, so.
We have time for another question. Okay.
Not a little advertisement. I'm gonna give a talk by the end of the day on approaching orchid morphology from arts, in particular from paper folding. So please don't miss it. It will be by the end of this day. Thank you.